Welcome to G-Fun Facts Online. Today we are leaving the safety of the Earth's atmosphere to talk about the single most critical engineering challenge for the future of our species. It is not the rocket engine. It is not the fuel source. It is the fragile, water-based biology of the human crew. When we look at science fiction, we see pristine ships and healthy crews. But the reality of deep space is a radiologically hostile environment that actively dismantles the machinery of life. For the last 50 years, human spaceflight has operated on a monitor and return paradigm. Astronauts on the International Space Station are shielded by Earth's magnetic field and are only hours away from a hospital. We collect their blood, freeze it, and study it months later. But as we pivot toward Mars, later is no longer an option. On a 3-year mission to the Red Planet, there is no abort, no resupply, and no hospital. This necessitates a complete paradigm shift in how we manage human health. We are talking about a concept called the DNA Sentinel. This is not a single device, but a converging ecosystem of technologies including nanopore sequencers, artificial intelligence, and synthetic biology foundries that function as a digital immune system for a deep space vessel. To understand why we need this, we have to look at the invisible war taking place at the molecular level in space. The primary enemy is galactic cosmic rays, or GCRs. These are atomic nuclei, stripped of their electrons, and accelerated to near light speed by supernova explosions. When a heavy ion like iron strikes a human cell, it creates a track of destruction. It can shatter the double helix of DNA, causing a double-strand break. While our cells have repair mechanisms, they are imperfect. In the chaos of a heavy ion strike, the cell might sew the wrong ends of DNA together, creating mutations that lead to cancer. Current dosimeters only measure radiation energy, but the DNA Sentinel is designed to measure the actual biological impact in real time. It is a biodosimeter. Furthermore, spaceflight induces immune dysregulation. More than half of astronauts shed latent viruses like herpes simplex or chickenpox because their T-cell function is blunted by the stress of spaceflight. Even the bacteria we bring with us behave differently. In microgravity, bacteria like Salmonella have been shown to become more virulent and form thicker, drug-resistant biofilms. The harmless flora in an astronaut's gut could evolve into a life-threatening pathogen. The DNA Sentinel is the answer to these threats. The core of this system is the ability to see biology, and the revolution here is nanopore sequencing. Historically, sequencing DNA required a room-sized lab. Today, thanks to devices like the Minion from Oxford Nanopore Technologies, it fits in the palm of your hand. Unlike traditional sequencing that uses light, nanopore sequencing uses electricity. Imagine a microscopic hole in a membrane. A current is passed through it. As a strand of DNA is ratcheted through this pore, each chemical base disrupts the current in a unique way. The device reads these electrical squiggles and translates them into genetic code. Astronaut Kate Rubins proved this works on the ISS in 2016. The DNA Sentinel takes this a step further with swab-to-sequencer technology. An astronaut can swab a vent or take a blood sample, inject it into a cartridge, and the machine autonomously extracts and sequences the DNA. This allows the ship to sniff for biological signatures 24/7. But raw data is just noise without context. This is where the brain of the Sentinel comes in, utilizing the concept of the digital twin. Every astronaut on a deep space mission will have a biological digital twin, a highly detailed virtual model of their physiology built from pre-flight data. The Sentinel feeds real-time omics data, transcriptomics, proteomics, and epigenetics into this digital model. If the sequencer detects elevated stress markers or inflammatory cytokines in an astronaut's saliva, the AI processes this against the digital twin. It might conclude that the commander is entering a state of chronic inflammatory fatigue with a high risk of viral reactivation days before any physical symptoms appear. This moves us from reactive medicine to predictive medicine. The system manages a trend, steering the astronaut back to health. Now, detection is useless without intervention. You cannot pack a pharmacy for a 3-year Mars mission because drugs expire and radiation degrades complex molecules. The solution is the astropharmacy. Instead of pills, the ship carries spores of Bacillus subtilis, a hardy bacterium genetically engineered to produce specific drugs. These spores can sit dormant for years. When the Sentinel detects that a crew member needs a specific antibiotic or a bone density booster, it wakes up the specific batch of bacteria, feeds them, and they synthesize the drug on demand. It is a living foundry. This extends to physical trauma as well. In the event of a burn or fracture, the Sentinel coordinates with 3D bioprinting systems, using bioinks derived from the astronaut's own stem cells, printing skin or bone grafts that eliminate the risk of rejection. The most futuristic aspect of this system involves CRISPR and gene therapy. Research like the Genes in Space 6 experiment has proven we can edit DNA in space. In a worst-case scenario, like a massive solar particle event where physical shielding is insufficient, the Sentinel could theoretically recommend a temporary gene therapy to upregulate natural radioprotective proteins, essentially creating genetic armor for the crew. Beyond protecting the crew, the DNA Sentinel has a duty to planetary protection. We cannot search for life on Mars if we contaminate the planet with Earth bacteria. Before an EVA, the Sentinel sequences the exterior of spacesuits to ensure sterilization protocols worked. Conversely, it protects Earth from backward contamination. If we bring samples back from Mars, the Sentinel acts as the gatekeeper, using AI to look for agnostic biosignatures, DNA patterns that do not match the Earth tree of life. If it finds something alien, it initiates a level 5 quarantine, ensuring the chain of contact is broken. Of course, this level of surveillance raises profound ethical questions. For the Sentinel to work, the crew must surrender total genetic privacy. They become transparent astronauts. The system knows their predisposition to diseases and their real-time psychological state. There is also the psychological burden of the quantified self. Imagine waking up to a dashboard telling you exactly how much your telomeres shortened overnight. It requires a delicate balance between transparency and morale. Ultimately, the DNA Sentinel is not science fiction. It is an inevitable engineering requirement. We cannot send 21st century organisms into a cosmic environment with 20th century medicine. This system represents the transition from visiting space to adapting to it. We are building a synthetic exoskeleton of data and biology. When humans finally step onto the red sands of Mars, they will be accompanied by the silent hum of the DNA Sentinel, the invisible guardian ensuring that the footprint they leave is human and that the home they return to is safe. That is the future of space travel, written in code and nucleotide bases.